I'm going to talk about our recent development of multifunctional optical coherence microscopy, which is for the investigation and assessment of the in vitro and ex vivo tissue. And I'm Yasuno from Computational Optics Group at the University of Tsukuba. Our application target is regenerative medicine, precision medicine, and drug discovery. For these applications, what we need to assess is in vitro, ex vivo, and animal tissues. And these tissues are actually living and it is thick. In addition, we are also interested in the functional imaging of these tissues. In addition, we are interested in the functional structure imaging, namely ultrastructure imaging. How we can do this? The main technology of us is optical coherence tomography, OCT. Although there are several advantages in OCT, there are also some open issues. For example, the resolution of OCT is still limited, and also there is a resolution depth of focus trade-off. And also OCT can only assess the tissue structure, not the function. But these limitations also can be overcome by using the advantages of OCT, which includes the three-dimensional holographic imaging, highly quantitative imaging, and high-speed imaging. Owing to these advantages, actually we can achieve the numerical pupil manipulation, ultrastructure imaging, and tissue dynamics imaging. So, the target of our multifunctional OCT microscopy project is achieving the high resolution and a long depth of focus simultaneously. And also, we would like to do the label-free, non-invasive longitudinal imaging of the tissue. And also, we would like to image the tissue dynamics and the functional structures. Let me summarize our targets. Our targets are actually four: resolution and depth of focus trade-off removal, an ultrastructure assessment, molecular specific imaging, and tissue dynamics imaging. The resolution, depth of focus trade-off, and the ultrastructure assessment can be performed by using the holographic signal processing. Molecular specific imaging can be done by using the polarization sensitive OCT, or in our specific case, it is a Jones matrix OCT. And the tissue dynamics imaging is going to be achieved by using the time sequential imaging and its statistical analysis. Let me clarify the measurement concept of our multifunctional optical coherence microscopy. The starting point of the measurement is the OCM or Jones matrix OCT hardware. By using it, we are going to have a three-dimensional complex tomography, and actually this is a three-dimensional holography. And then we are going to apply several types of signal processing, and each of the signal processing corresponds to each of the measurement functions. But some of the processing requires a highly stabilized phase of the three-dimensional complex tomography, and this high stability of the phase is going to be achieved by a newly developed phase stabilization method. And finally, after applying the signal processing, we are going to have multifunctional OCM images. At the basement of the measurement, we built two types of OCT. The one is a spectrodynamic OCT with an 840 nanometer probe. It has a relatively high resolution of 3.6 microns in depth and 4.8 microns in lateral, and it has a measurement speed of 80,000 A lines per second. The second system is a swept source Jones matrix OCT with 1.3 micrometer probe. It has a relatively lower resolution, as the depth resolution is 14 microns and the lateral resolution is 90 microns, and the measurement speed is 50,000 A lines per second. But this system is characterized by its capability of measuring the birefringence and the degree of polarization uniformity. Let me explain the measurement function one by one. The first measurement function is holographic refocusing. Our holographic refocusing is based on forward propagation model. The starting point of this method is a three-dimensional complex field distribution measured by the OCT. We extracted an en-face distribution of a complex electric field and Fourier transform it. By this, we are going to have the complex field distribution at the pupil plane, and then we are going to add the phase function which mimics the additional focusing lens, and then inversely Fourier transform it into the sample domain. By this operation, we can change the depth location of the focus after the acquisition. By applying this method to all of the depth, we can have OCT volume which is in focus throughout all of the depth. The concept of the holographic refocusing itself is somehow old, but its implementation for the real sample is a little bit difficult, and especially it is because of the depth-independent phase error which is occurred during the measurement. To estimate it and remove it, we built a new algorithm. The basic strategy of this algorithm is to first to compute the vectorial gradient of the phase error, or say bulk phase error, vectorial gradient of the bulk phase error, and then apply the line integration to estimate the bulk phase itself. However, this line integration is always associated with the accumulation of the error, so this very simple method couldn't have a high accuracy. How we can overcome it? To overcome it, we built a second algorithm, so-called as a smart integration path. Let's assume we want to estimate the phase of the center pixel, so-called as a target pixel, from the phase of corner pixel, blue in this diagram, or so-called as a source pixel. From the source pixel to the target pixel, we can think of 4 integration paths. And actually, we did the integration along these 4 paths and then apply the averaging in complex for the results of these 4 paths, and which will give us a final estimation of the phase of the target pixel. The algorithm itself is a little bit more elaborated, and if you're interested in, please refer to our recent publication in Biomedical Optics Express. Due to the bulk phase error correction, now we can achieve highly accurate refocusing. Here is an example of the chicken breast muscle. In the original image, there is some structure in focus, but a structure becomes blurred and blurred as the defocus increases. But once we apply the bulk phase error correction and computational refocus, we see a very fine structure even with the defocus. At the 640 micrometer defocus, we still have a very fine structure of the muscle fiber. Let's go to the second measurement function, quantitative 3-dimensional polarization imaging achieved by Jones matrix OCT. Jones matrix OCT is a hardware extension of the optical coherence tomography, and which measures 4 images associated with 2 incident polarization and 2 output polarization simultaneously. And these 4 images form Jones matrix of the sample. Once we measure the Jones matrix of the sample, we can derive several clinically useful images from the Jones matrix. It can include a high-sensitive OCT and also local scattering. By applying the phase analysis, we can have a Doppler OCT, and by applying the correlation analysis, we are going to have a flow contrast, and which can be used to generate the OCTA image. By applying the eigenvalue analysis, we are going to have a birefringence image, and it is known to be sensitive to collagen. By computing the polarization variance, we are going to have the image so-called as degree of polarization uniformity, or DOPU, and it is known to be sensitive to melanin. The concept of the Jones matrix OCT is not new, but here in this project, we combine it with the computational refocusing. Here is an example of a chicken breast muscle. In the M-fast OCT image, the structure is totally blurred. Once we turn on the polarization sensitivity, we see a birefringence image. Here in this image, some region shows the green appearance, namely modulated high birefringence. And in some region which appeared with red, it's an indicator of extremely high birefringence. But once we turn on the holographic refocusing, the situation becomes a little bit different. By zooming it up, for example, here we see a very fine structure of the muscle fiber, and this muscle fiber shows a modulated high birefringence in green. And here in these regions, we see a blue appearance outside of the muscle fiber. It is an indicator of low birefringence. In addition, the high birefringence, say red stripes in the original image is gone. So this means these high birefringences are actually not true, but an artifact caused by the defocusing. Let's go to the measurement function 3, is the tissue dynamics imaging. The tissue dynamics imaging is a combination of the time sequential OCT measurement and its statistical analysis. For the statistical analysis, we developed two algorithms. The first one is log intensity variance, or LIV in short. It assesses the magnitude of the fluctuation of the OCT signal along the time. So it might associate it with the magnitude of an intracellular motion of the sample. The second quantity we computed is the OCT correlation decay speed, OCDS. It's a decay speed of the autocorrelation function of the OCT signal along the depth. So if the speed is fast, the decay speed is fast, it indicates the fast movement in a tissue or in a cell. We examine the anti-cancer drug response of tumor spheroids by the dynamic OCT imaging. The spheroids of human breast cancer cell were cultured and anti-cancer drug paclitaxel was applied. In the OCT cross-section, we only see the spheroid morphology and cannot identify the active region of the spheroid. By LIV, we can identify the region with large magnitude of intracellular motion. For the low concentration of the drug, the outer part of the spheroid is active. The inner part is not active because of lack of nutrients. This situation is reversed with the high concentration of the drug. Here the outer part of the spheroid is affected by the drug and became inactive. OCDS showed slightly different appearance. For the low drug concentration, the inner part of the spheroid showed high OCDS signal. It may indicate the activity of the necrosis. So let's go to the final measurement function. It is computational directional imaging. The idea of the computational directional imaging is somehow similar to the holographic refocusing. Here again, we extract the complex and fast OCT signal and then applying the two-dimensional Fourier transform to obtain the electric field distribution at the pupil plane. And in this case, we are going to apply the intensity directional numerical mask to the pupil plane and then inversely Fourier transform it into the sample domain. By properly selecting the intensity mask, we can visualize the sub-resolution structure of the sample. Here is an example of a chicken breast muscle. This image is a combination of two directional images as indicated in the inset. With this direction set, we see a clear stripe pattern along the muscle fiber. The second image is also a combination of two directional images, but direction is perpendicular to the muscle fiber structure. This configuration reveals the crimped structure of the muscle. In the cross-sectional image, we see a clear boundary between two tissue types. Also in the en-face images, we see the clear boundary. But in a standard OCT image, this boundary is unclear. Once we see the pseudo-color computational directional image, it is easy to identify the boundary. Let me summarize my talk. In this project, we are working for Multicontrast OCT microscope, and its core is the OCT microscope hardware. OCT microscope is quantitative, has high speed, and can measure the complex electric field. By exploiting these properties, we achieve the four measurement functions, including dynamics imaging, polarization imaging, holographic refocusing, and outer structure imaging. These measurement functions would be useful for the assessment of ex vivo in vitro tissues and can be used for precision and regenerative medicine and also drug discovery. Thank you for your attention.